Frank Myers. He's going to talk about an integrated microsystem for electrophysiology-activated cell sorting. Frank is a fifth-year PhD student. He's going to graduate next May, so he may be looking for a job.
All right. Thanks, Liwei. So yeah, I'm going to talk to you about a new technology that we're developing in our lab for cell sorting. That rather than looking at a protein or a genetic marker for cell phenotype, which is typically done for cell sorting, we're looking at a cell's behavior, namely its electrophysiology. So electrophysiology is really a label-free indicator of cell phenotypes. Certain cell types, namely muscle cells and neuronal cells, express voltage-gated ion channels in their cell membranes that upon stimulation, upon electrical stimulation, these ion channels transiently open and allow an ion current to flow across the cell membrane. And the shape of that ion current through time, that signal, is unique for different cell types. So we can use this to distinguish different kinds of cells. And we're particularly interested in this because for a lot of cell types that are relevant for stem cell therapies, the cells are electrically excitable. So cardiomyocytes for heart transplant therapies, smooth muscle for vascular transplant therapies, and so forth. So we're interested in using this as a way of purifying cells out from stem cell-derived populations for therapy. And during stem cell differentiation, let's see, sorry, this is automatically going to start. So during stem cell differentiation, we get a variety of different cell types. And this is cardiomyocyte differentiation, so we're specifically trying to coerce these human pluripotent stem cells into heart muscle cells. And as you can see, we get a few colonies that are beating. Those are cardiomyocytes. They're spontaneously beating. If we electrically stimulate them, they'll respond. And we get a lot of other cell types in there that are non-responsive. They're not cell types we want. For our purposes, they're contamination. So we'd like to get rid of those. And specifically, we're interested in isolating cardiomyocytes which look like ventricular cells, because in typical heart infarction, the injury occurs in the ventricles of the heart. So that's the tissue you'd like to replace, and you'd like to find cells that resemble that tissue. And so electrophysiology can give us some insight into what kind of cardiomyocytes we have. And within a given stem cell-derived population, there are a variety of different cardiomyocyte phenotypes: atrial, ventricular, pacemaker type, as well as smooth muscle cells. And the heart is really a mosaic of these different cell phenotypes. So if we can specifically isolate cells which resemble ventricular phenotypes, then that's what we want. Conventional cell purification, generally we think of FACS, fluorescence-activated cell sorting, in which a fluorescently conjugated antibody is targeted to a protein on the cell's surface, and that uniquely identifies a certain cell type. Unfortunately, for cardiomyocytes, there are not good surface markers to target, and even if there were, the labeling molecule can interfere with the performance of a tissue graft. So that's not ideal. And we can also do genetic methods to select for different cell types, but any kind of genetic modification of cells has a substantial tumorigenic risk, and that's generally to be avoided for therapy. And finally, there are centrifugation methods which rely on different cell densities to determine cell phenotype, but these are very nonspecific. So we think that electrophysiology, being label-free, non-genetic, and also with potentially a very low false positive rate, because for non-cardiomyocytes, it's very unlikely that they would express the host of voltage-gated ion channels on their surface to make them look like cardiomyocytes. So potentially a very low false positive rate. And this is a functional assessment. You're asking the cell to perform a task. You're stimulating it and seeing if it responds. And that task is intimately coupled with the kind of task you want it to perform in a tissue graft, namely contract in response to electrical signals in the heart and do that in an organized, predictable manner. So that's something that we're getting at here with this cell sorting technique. This is the kind of conventional view of, or the biologist view of cell membrane physiology. Lots of ion channels which provide these transmembrane signals, ion pumps which maintain ion gradients, Being an engineer, we like to simplify things down. So this is the electrical engineer's view of the cell membrane, the Hodge-Huxley model. And basically, the cell membrane can be thought of as a variety of voltage-gated conductances for different ions. And so these are functions of the transmembrane voltage as well as time. So the gating kinetics for different ion channels, and there are many, are unique. And so depending on the panel of ion channels that a cell is expressing, you will get a different aggregate signal. So you'll have potassium currents, calcium currents, sodium currents going in and out of the cell, but they'll all add up to this sort of dotted line here in this cartoon of an aggregate current. And there's also an associated transmembrane voltage. This is a capacitor, so the transmembrane voltage is a derivative of that transmembrane current. And there are a couple of ways to get at this signal. Patch clamping is the most common, and that's done in electrophysiology studies. And in that case, you're actually puncturing a hole in the membrane with a pipette, and you're measuring the transmembrane potential directly. These are called action potentials, and they're in the millivolt range. They're very easy to measure. Microelectrode recordings, on the other hand, rely on they don't puncture the cell membrane. They rely on the extracellular ohmic potential drop due to that transmembrane current. And so they're much weaker. These are called field potentials, and they're in the microvolt range. But they're non-invasive, and that's what we use in the cell sorter. So our device is basically a microfluidic flow cell with electrodes on the floor. And this is how it works. The cells are introduced through a central inlet and then hydrodynamically focused with these flanking sheath flows. And when they pass into our electrode detection region, we see a drop in impedance on these two small electrodes, and we stop the flow. And then actually, I should say that this is wrong. It should be an increase in impedance, right? Obviously, because there's an obstruction, and this is backwards. I corrected it on the poster. But so then, once the flow is stopped, a stimulus pulse is delivered through these two large rectangular electrodes, and then the differential signal is measured in the vicinity of the cell. Now, this geometry is important because we're delivering about 10 volts across those two large electrodes in order to get enough current to stimulate the cell. And we want to measure signals on the order of 10 microvolts. So you can imagine that's a substantial stimulus artifact that we have to deal with. And one of the ways we do that is by placing these electrodes on an equipotential line in the stimulus field so that the stimulus is essentially common mode and rejected. We analyze that signal and then open up one of several valves to sort the cells. So this is our prototype. This is just showing the electrodes fabricated. Electrodes are platinum with a silicon nitride passivation layer, and then the flow channel is made of PDMS. And that's placed into our instrumentation here. This is the chip in the middle. And then, of course, take another step back. The syringe pump is a syringe pump for delivering the cells into the device, and then outlet reservoirs, which have valves on them for selecting the outlet. And then the whole thing is automated with LabVIEW. So, as I mentioned, for stimulus artifact suppression, this orthogonal geometry is very important. We get a substantial reduction in the artifact. This is a stimulus artifact from a 1 milliamp pulse. And you can see if I make a single-ended recording, it just saturates the amplifier for days. If I make this differential measurement, then it substantially reduces it. And I can further reduce it by isolating the stimulus generator, so there's no common ground between the recording amp and the stimulator, and I get pretty nice reduction here in the artifact. And finally, we actually coat these electrodes with platinum black, which reduces their impedance, and that's good for reducing the noise and increasing our SNR on our measurement end, but it's also good for reducing the stimulus artifact. And then finally, the remaining artifact is removed in software. So for stimulation, between these two large rectangular electrodes, this is basically what it looks like. Current is injected through the electrodes, and the membrane is essentially a capacitance, so the current through that membrane causes it to charge up, and at a certain point reaches the stimulus threshold. And one of the nice things about field potential measurements, if you talk to people that do a lot of electrophysiology, field stimulation is considered like a hammer over the head on the cell. It's very nonspecific. It's kind of brute force. But because we are confining, or we are determining the geometry here. We've designed it. We know exactly how the current is being distributed through the cell, and so we have a lot of control over how the cell is being stimulated, and we can make basically if the channel is 5 times larger, we need 5 times more current to get the same current density for stimulation. So it's very deterministic because the geometry is well defined. For stimulation, we're looking at the 2 small electrodes there in the middle. And again, we're looking at the ohmic drop in the resistive medium surrounding the cell due to the transmembrane current. And so it looks something like that, the potential field around the cell. And one of the nice things here about doing this in microfluidics is that by shrinking that channel cross-section down, we actually get a boost in the signal amplitude because that increases, reducing the cross-sectional area, increases the effective resistance through which that transmembrane current has to go. So this is what conventional microelectrode recording looks like, where the cell is adhered on the electrode. It's an intimate contact with the electrode. And in our device, if the cell is just floating, it's in suspension, the amplitude is lower. But if we tune the geometry so that the cells are filling most of the channel, we can recover that amplitude and we can get SNRs of the same, roughly equivalent to microelectrode recordings, which is nice. And so this is just showing you a sorting experiment. The first, this is a cluster of cells, so these are probably about 1,000 cells in an aggregate. And you're going to see one cluster come through and not respond to stimulation. And then the next one will respond, and they'll be sorted out of different outlets accordingly. So cells flow in, there's a blip in impedance across these electrodes, a stimulus pulse train is applied, the stimulus artifact is filtered out, and nothing is recovered, so the cells are sorted out of outlet C over here. In this case, again, we get a blip of an impedance, flow is stopped, the cell is stimulated, and it responds. Responds here, and it also spontaneously beats here. And by varying the stimulus parameters, you can get at different physiological parameters. You can look at refractory intervals, you can look at stimulus threshold by applying a staircase and so forth. So it's a parametric analysis, which is kind of cool. We can also look at spontaneous signals from cells. And typically for a therapy, you wouldn't want cells that are spontaneously beating because they're going to, your graft is not supposed to be the one that's driving the pacing of the heart, right? So you want them to respond to stimulus, but you don't want them to spontaneously beat. And we can select for that with this cell sorter. So we looked at using this to distinguish undifferentiated undifferentiated stem cells from cardiomyocytes derived from the same stem cells. And cardiomyocytes produce these nice evoked field potentials upon stimulation. They also produce spontaneous field potentials. Undifferentiated cells do not produce these spontaneous field potentials and do not produce signals in response to stimulus. And if you look at these signals, they're rich with phenotypic information. All of these amplitudes and timing parameters mean something in terms of the ion channels that the cell's expressing. So our vision for this ultimately is to parallelize it. It's a fairly slow measurement on the order of hundreds of milliseconds to seconds, but the electronics here are all sort of Walkman level. There's nothing really inhibiting the scale up of this, and so you could imagine a chip with parallel sorting channels, hundreds or thousands of parallel sorting channels, to get the throughput that you would need for analysis or for therapy. And so, in summary, I hope that I have convinced you that electrophysiology could be a useful contrast signal for label-free, non-genetic, and a functional assessment of cell phenotype. And our cell sorting technology here can distinguish differentiated cardiomyocytes from their undifferentiated counterparts. And finally, I just kind of want to throw this out there. I mean, if stem cell biology translates into therapy, like a lot of people think it will, then we're going to have a new industry emerge in which the product is a mammalian cell. And we don't really have a lot of technologies out there for producing large industrial scale quantities of mammalian cells with consistency and quality control and all of that. So I think that microsystems, because many of the factors influencing a cell's development and growth are microscale, the chemical environment, cell contact mediated signaling. I think that microsystems have a role to play in terms of the industrial scale production of mammalian cells, and I think for tissue replacement therapies, that's definitely pretty cool. So I'll take any questions. Thanks.
Any question for the speaker?
Sure.
Here's one.
So very, very exciting work. I had a question. So when you showed the signal after extracting the background, it wasn't at all visible, I think, to me, that signal was there in the raw signal. And so, like, what sort of actually, how hard is it to extract that?
This signal here?
Maybe if you go back, let's see.
The video?
You were showing a little bit further. Yeah, exactly. So you show after artifact removal, like in that blue pulse train, it's not obvious to me that there's anything different about that third pulse from all the others. Is it just that we're not at the right scale here, or? Right.
Yeah. I mean, the artifact is obscuring it, and so it's actually it's in there, but you need to, I guess. Let's see. Maybe what I should show you is more of a close-up view of. Yeah, these aren't particularly good either, but I mean, this artifact is obscuring that signal. Maybe if you look closely at this one, actually, yeah, because this one, you notice how it dips and then recovers here, whereas here it doesn't.
Okay.
Do you see the difference?
Yeah.
Because so every one of these it goes up and then back down, and there's a motion artifact associated with that contraction. The reason this one responds and that this one doesn't, and the same thing with this one, and it looks like this one responds, is because this stimulation occurs after the refractory interval. So there's a minimum amount of time that the cell has to have in order to regain its transmembrane ion gradient. And so this stimulus happens to occur after that amount of time, whereas this one doesn't. So yeah, I agree. It's kind of hard to see there, but trust me, it's there. I didn't just Photoshop it in. Any other questions?
Okay, let's thank Aman again. Next speaker will be